Hello everyone, I am Gayatri. Today I am going to present a seminar on the topic Pathogenesis of tumors associated with Epstein-Barr virus. Let's look into the overview of the pathogenesis. Now coming to the introduction to the Epstein-Barr virus associated tumors. This presentation explores the biology of Epstein-Barr virus and its role in tumor development, aiming to enhance the understanding of associated pathogenesis, tumor types and clinical implications. Before going into the pathogenesis, let's look the virology of Epstein-Barr virus. Initially, there is an envelope, which is the outer layer. It is a lipid membrane taken from the host cell. It contains envelope proteins, which are also called as spikes. These helps the virus attach to the B cells via CD21 receptor. Inner to the envelope, we can see tegument. Tegument is nothing but the middle protein layer, which contains the viral proteins and enzymes. This helps in the kickstart infection once the Epstein-Barr virus enters the cell. Inner to the tegument, there is a nucleocapsid, which is a protein shell which is icosahedral in symmetry. This protects the viral genetic material. The viral genome core, which is inner to the nucleocapsid, is double-stranded DNA. It contains the genes like LMP1, EBNA2, those are responsible for B cell proliferation, immune invasion, and oncogenesis. Epstein-Barr virus is also called as human herpesvirus 4. Now, let's look detail into the genome of Epstein-Barr virus. Inside the host nucleus, the DNA becomes circular. This is the behavior of the genome of the Epstein-Barr virus and the cell. The circular DNA is also called as episome. This persists during the latent infection. Now, let's look into the latent genes, which are most important. LMP1 or latent membrane protein 1. This acts like CD40 and it activates NF-kB pathway and JAK-STAT pathway. NF-kB pathway is nothing but nuclear factor kappa B pathway, and the JAK-STAT pathway is Janus kinase signal transducer activator of transcription. This is a signal transduction pathway used by growth factors and cytokines. It connects the cell surface receptor and nucleus. Now, the effects of latent genes are B cell proliferation and anti-apoptosis via increased BCL2. Now comes the EBNA or Epstein-Barr nuclear antigens. There are two types, which are EBNA1 and EBNA2. EBNA1 maintains the viral genome in the host cell. EBNA2 mimics the Notch signaling. Notch signaling is nothing but the cell-to-cell communication pathway controlling cell differentiation, proliferation, and the fate decision of the cell. Now, what are the functions of EBNA? EBNA activates cyclin D and the proto-oncogene. Cyclin D is a transition control from the G1 to the S phase of cell division. Also, it is a proto-oncogene product which promotes the proliferation. Now, the third one, Epstein-Barr virus encoded RNAs or EBERs. Those are non-coding RNAs that helps in immune invasion and cell survival. Now the fourth one is viral interleukin 10. This mimics the interleukin 10 seen in humans. Also, they are acute inflammatory cytokines that suppress the immune response. Now, let's see the Epstein-Barr virus infection cycle. First, As in the picture, you can see the Epstein-Barr virus enters to the body of the human and it infects mainly the oropharynx. Infects the oropharynx and the cells such as epithelial cells and then the B cells. In the B cells, the virus binds to the CD21 receptors using glycoprotein 350 antigen. Also, entry into the B cell starts the infection. Now, there are two phases in the life cycle which are lytic and latent phase. Now, let's discuss about the lytic phase, which is the phase of active replication. Here, the viral DNA replicates and new virions are formed. Cell may lyse and the virus spread. Lytic phase is seen in both epithelial cells and B cells. But the latent infection, here the virus becomes latent only inside the B cell. No full viral production is seen and express limited proteins such as eBNA, LMP1, LMP2 and EBER as we have already discussed. And latent infection promotes long-term survival. Here, the B cell proliferation and immune response can be seen. The infected B cells proliferate and infectious mononucleosis symptoms are present at this stage. Now, the establishment of memory B cells and the reactivation under stress or immune suppression can be seen. There is a conversion of latent to the lytic phase in these conditions. We'll see detail as we have already discussed. First one is entry into the host, that is spreads via saliva. It is also called the kissing disease. Virus binds via glycoprotein 350 to CD21 receptors on the B cells. The fusion and entry. The fusion of the viral envelope with the cell membrane nucleocapsid enters the cytoplasm and then to the nucleus. Genome delivery and circularization. The viral DNA enters the nucleus. The linear DNA becomes circular episome. This is the key for latency. Two possible pathways as we have already discussed, the lytic cycle and the latent cycle. The lytic cycle is seen in epithelial cells as to the B cells. There is the replication and the cell lysis and the viral release. In the latent cycle, which happens in the B cells, that is limited proteins are expressed like eBNA, LMP expression, which lead to the lifelong persistence and then reactivation. Now, let's look into the type of latency. Latency 1, latency 2 and latency 3. Latency 1, the viral protein expressed are EBNA1 and EBER. The key feature is there is only a minimal expression of these proteins and the associated disease as Burkitt's lymphoma. In latency type 2, the viral proteins expressed are EBNA1, LMP1, LMT2 and EBER. There is a moderate expression and associated diseases varies like nasopharyngeal carcinoma, Hodgkin lymphoma. Now coming to the latency 3, EBNA 1, 2, 3 and LMP 1, 2 and EBER. These are the viral proteins expressed here. The key feature is a full expression of these proteins and the associated diseases are immunosuppressed B cell lymphomas. Now let's look into the molecular mechanism which happens inside the cell. Epstein-Barr virus induces cellular transformation through latent gene products that promote proliferation, inhibit apoptosis and invade immune response. This is the overview of pathogenesis. The normal epithelial cells undergo genetic alterations and this leads to the mild dysplasia. With the Epstein-Barr viral infection, there are the expression of several RNAs like small RNAs and mRNAs. Those are EBERs and BARTs. Those promote the growth and interferon induction, maintain the viral latency, apoptosis resistance and immune evasion. From the mild dysplasia, The Epstein-Barr virus infection leads to the severe dysplasia. Now, the severe dysplasia with the accumulation of genetic and epigenetic changes lead to carcinoma in situ. And again, with the genetic and epigenetic changes, it leads to invasive carcinoma. Not only that, the Epstein-Barr viral lateral gene expressions like LMP1, LMP2 and EBNA1 also result in the transformation of carcinoma in situ to the invasive carcinoma. This is done by the mechanism of growth promotion, anti-apoptosis, invasion and metastasis promotion, immune evasion, suppression of cell differentiation, metastasis promotion, episome maintenance, etc. Now we'll look into the pathogenesis. Hijacking the host signaling pathways. This is the main mechanism which lead to the pathogenesis. Epstein-Barr virus promotes the B cell proliferation by mimicking normal signaling. The key viral proteins, as we have already discussed, the latent membrane protein 1 that acts as an oncogene and functions like a constitutively active CD40 receptor. It activates the nuclear factor kappa B pathway and the JAK-STAT pathway. The effects are increased B cell survival and increased proliferation even without the T cell help. It also prevents apoptosis via BCL2 activation. Now, the LMP1 serves in the survival, proliferation and anti-apoptosis. Now coming to the next protein, which is EBNA2. EBNA2 mimics the Notch receptor signaling. Now, what is Notch receptor signaling? It is nothing but the cell-to-cell communication pathway controlling the cell differentiation, proliferation and fate decision of the cell. Also, it activates the host genes like cyclin D, SRC family proto-oncogenes. Promotes the cell cycle progression and proliferation. Now, what is the mechanism adopted by the host in response to the Epstein-Barr virus? Let's look the interaction with the host. Epstein-Barr virus alters the host cell signaling pathways to promote survival, proliferation and immune evasion in infected cells. Now the immune response and outcome. In immunocompetent individuals, the Epstein-Barr viral driven B cell proliferation is always controlled. The outcomes are asymptomatic infection or infectious mononucleosis, which is self-limited. Infectious mononucleosis will be dealt in the later slides. Immune evasion is critical for cancer development. The genetic changes in the Epstein-Barr viral associated tumors include chromosomal translocations and DNA methylation patterns, significantly influencing tumorigenesis and contributing to the distinct pathology of these malignancies. Now let's see what are the tumor types associated with Epstein-Barr virus. We'll discuss one by one. The first one is Burkitt's lymphoma and its subtypes, and Hodgkin lymphoma with Epstein-Barr viral association. Next comes the nasopharyngeal carcinoma, and the last gastric carcinoma and its variants. We'll come first to the Burkitt's lymphoma. Burkitt lymphoma is nothing but the tumor of the B lymphocytes. These are common in Central Africa and New Guinea. It is the most common childhood tumor seen in these areas. Now let's look the association with Epstein-Barr virus. 90% of the African cases contain Epstein-Barr viral genome. 100% shows increased antibodies against the viral capsid antigen. Antibody levels correlate with the tumor risk. Let's come to the pathology of Burkitt lymphoma. The key genetic event is MYC oncogene activation via translocation. MYC oncogene is present in the chromosome 8. It is a nuclear transcription factor. MYC protein is a nuclear transcription factor and MYC gene is an oncogene, and it leads to the increased MYC protein. As it is a transcription factor, there is uncontrolled proliferation, and there are some important observations which need to be forced in Burkitt lymphoma. Like Epstein-Barr virus infection is ubiquitous worldwide, but only 15 to 20% of the non-African cases have Epstein-Barr virus. This indicates that the cofactors are required for the pathogenesis of Burkitt lymphoma. Now let's see the role of cofactors. Example, chronic malaria. Chronic malaria causes the immune suppression and it allows the sustained B cell proliferation. Thus, the pathology of Burkitt lymphoma or the pathogenesis of Burkitt lymphoma becomes easier. So this is the role of cofactor. Now let's see the overview. Epstein-Barr virus infects the cell and there is a latent infection and later leading to the polyclonal B cell expansion. Immune system controls the most cells, which are immune system mainly the cytotoxic lymphocytes. These control most the cells, but some cells evade the immunity, then acquire the MYC translocation as dealt earlier, and additional mutations may occur. The monoclonal malignant proliferation leads to the Burkitt lymphoma. This is the important conclusion. Epstein-Barr virus is not directly oncogenic in Burkitt lymphoma. Instead, it acts as a polyclonal B cell mitogen. What is a mitogen? Mitogen is nothing but a factor that promotes the cell differentiation. Here, it is B cell differentiation, and it creates a setting for mutations like MYC, as seen in the translocation. The final transformation requires 8-14 translocation and additional mutations. The pathology features are important. Burkitt lymphoma exhibits a characteristic starry sky appearance, which is marked by high proliferation of atypical lymphoid cells. The starry sky appearance. Now let's see the pathological features. Burkitt lymphoma exhibits a characteristic starry sky appearance, which is marked by a high proliferation of atypical lymphoid cells. The starry sky appearance is characterized by scattered macrophages known as tingible body macrophages within a background of closely packed atypical lymphoid cells. Burkitt lymphoma typically expresses CD10 and BCL6, indicating an activated germinal center phenotype, which aids in the diagnosis and understanding of its pathophysiology. You can see the histopathological feature in the picture given. Now coming to the Hodgkin lymphoma. The Hodgkin lymphoma is a B cell derived malignancy. It is characterized by Reed-Steinberg cells and a background of mixed inflammatory cells that contain lymphocytes, eosinophils, and plasma cells. Epstein-Barr virus is implicated in a subset of Hodgkin lymphoma cases that varies by subtype and geography. Now let's see the association rates with Epstein-Barr virus. The mixed cellularity HL, there is a 70% positive Epstein-Barr virus cases. Lymphocyte depleted HL, there is a high Epstein-Barr virus association. In nodular sclerosis HL, there is only 10 to 40% association with Epstein-Barr virus. And nodular lymphocyte predominant HL or Hodgkin lymphoma, usually the Epstein-Barr virus is absent or there is no association with Epstein-Barr virus. Now let's see the cell of origin. The RS cells arise from the germinal center or post-germinal center B cells. These cells are Lowe's typical B cell markers. They gain abnormal phenotype due to genetic and viral effects. Now let's see the molecular pathogenesis of Hodgkin lymphoma. The role of LMP1, which is a key oncoprotein, and the pathogenesis, which mimics the CD40 receptors and activates NF-κB and JAK-STAT pathway. We already knew this. Now let's see the effects. The effects are, as usual, prevents apoptosis, promotes the survival of RS cells, and induce the cytokine production. The central event, which is the nuclear factor kappa B activation, it is seen in almost all Hodgkin lymphoma cases, whether there is an association with Epstein-Barr virus or non-associated cases. In both cases, we can see the central event, that is NF-κB activation. In Epstein-Barr virus positive cases, that is driven by LMP1. In Epstein-Barr viral negative cases, this is due to genetic mutations, and this lead to the cell survival and resistance to apoptosis. Now, LMP2 function. It mimics the B cell receptor signaling and prevents the apoptosis of defective B cells. Now coming to EBNA1, this maintains the viral genome. Now let's see what is Reed-Sternberg cell. Let's see the morphology. Those are large cells with a bilobed nucleus. This is a peculiar feature seen in Hodgkin lymphoma. And there is a prominent nucleoli. So we call it as owl's eye. Now coming to the immunophenotype, there is CD15 and CD30 presence. These are markers. Also, there is a weak B cell markers, which is CD20, which is variable. Now, the tumor microenvironment. The RS cells are few in number. Majority are the reactive inflammatory cells because RS cells secrete cytokines that recruit inflammatory cells. Now coming to the pathology of Hodgkin lymphoma. As we already discussed, Reed-Sternberg cells. Those are large atypical cells characteristic of Hodgkin lymphoma, typically found in an inflammatory background, often surrounded by reactive lymphocytes and histiocytes. Coming to the immunohistochemistry, immunohistochemical profiling of the Hodgkin lymphoma reveals CD30 and CD15 positivity, which are vital for diagnosis, helping to distinguish it from other lymphoproliferative disorders. We have already discussed the histopathological characters, that is Reed-Sternberg cells. You can see the owl eye appearance and the histopathological slide. Now coming to the next session, that is Epstein-Barr virus in immunosuppressed patients. Examples: AIDS and the transplant recipients. The transplant recipients will be on immunosuppressive therapy. So, there is a prominent suppression of the immune responses. As you all know, AIDS is an immunosuppressive disorder. And now let's see the features. The Epstein-Barr virus plays a major direct role in these patients. The tumors are often multifocal and occur in extranodal sites like gut, central nervous system. Initially, it is polyclonal and later it progresses to monoclonal. Now let's see the difference from Burkitt lymphoma. Here, there is an expression of LMP1, EBNA2. Usually, no MYC translocation. MYC translocation is seen in Burkitt lymphoma. And now let's see the reversibility. The tumors may regress if the immune function improves, such as stopping the immunosuppressive drugs can reverse the condition. Now coming to the next tumor associated with Epstein-Barr virus, which is the nasopharyngeal carcinoma. Let's see the epidemiology first. It is common in southern China and parts of Africa and Inuit populations. The association rates: 100% of the tumors contain Epstein-Barr virus. The viral genome is clonal and infection occurs before the tumor develops. Now let's see the immunological findings. High antibody titers against the viral capsid antigens are seen. That is, early IgA antibodies are prominent. The molecular mechanisms are: tumor cell suppress the LMP1 and activates NF-κB pathway. The effects of NF-κB activation here is that it upregulates the VEGF, which leads to angiogenesis, and FGF2, which leads to the growth, and MMP9, which lead to the invasion, and COX2, always inflammation and tumor progression. Now let's see the pathology of nasopharyngeal carcinoma. Carcinoma is nothing but undifferentiated carcinoma shows a distinctive lymphoid stroma characterized by scattered tumor cells within a background of lymphocytes contributing to the tumor's unique pathological features. EBER in situ hybridization confirms the presence of Epstein-Barr virus within tumor cells, highlighting its significance in the pathogenesis of nasopharyngeal carcinoma and aiding in definitive diagnosis. Now let's see Epstein-Barr virus associated gastric carcinoma. Here, approximately 10% of the gastric cancers are associated with Epstein-Barr virus, highlighting its significance in the pathogenesis of this malignancy. The latency 1 is exhibited here in the gastric carcinoma, where several selective genes are expressed, contributing to the tumor development and progression. Now let's see the pathology of Epstein-Barr viral associated gastric carcinoma. There is lymphoepithelioma-like carcinoma. This carcinoma subtype exhibits distinctive features resembling undifferentiated carcinoma with prominent lymphoid stroma, making it crucial for accurate diagnosis through histopathological evaluation. And EBER positivity. EBER in situ hybridization confirms the EBV presence, aiding in the diagnostic process by highlighting infected cells within the tumor microenvironment and supporting the role of EBV in carcinogenesis. Histological variants. The Epstein-Barr viral gastric carcinoma features, that is lymphoepithelioma-like carcinoma subtype with the distinct diagnostic markers including EBER positivity for effective detection. Now let's see the other Epstein-Barr virus associated disorders, which are post-transplant lymphoproliferative disorder, and we have already discussed about the immunocompromised patients and the associated with a latent Epstein-Barr virus infection and presence as lymphadenopathy or organ infiltration. And the treatment include antiviral therapy and immunotherapy additionally. HIV associated lymphomas. Increased risk in HIV positive individuals associated with high Epstein-Barr virus load, and so common type includes primary CNS lymphoma. Treatment typically involves antiretroviral therapy and multidisciplinary approach essential for management. Now let's see the diagnostic techniques. The major diagnostic approaches are dealt here. And the first one is the serological test, which is the most common or the first line approach. Antibodies against the Epstein-Barr viral antigens are targeted here. The viral capsid antigen IgM is seen in acute infection, and viral capsid antigen IgG is seen in past or current infection. The Epstein-Barr viral nuclear antigen is the indicative of past infection, and early antigen an indicative of active replication. The multiple antibody patterns, up to 32 combinations, help determine infection stage. The common tests are ELISA, which is enzyme-linked immunosorbent assay, and heterophile antibody test, which is also called as monospot test. Now let's see the advantages of serological test. These are widely available and also cost effective. Now let's see the limitations. These may give false positive or negative results, and also they are less useful in immunocompromised patients. Now let's see the second method, which is a gold standard for sensitivity. These are molecular methods. The techniques are polymerase chain reaction, quantitative polymerase chain reaction, nucleic acid amplification test. What they detect? They detect the Epstein-Barr viral DNA. That is a viral load in blood, tissue and cerebrospinal fluid. The research findings show that PCR sensitivity is 96%, whereas the specificity rises to 100%. Viral load helps to distinguish between the latent infection and active disease. The advantages are highly sensitive and specific, and quantitative monitoring is possible here. Limitations are obviously the cost, and it requires specialized labs. Now let's see into the modern or advanced techniques which are emerging in recent researches. The new technologies include CRISPR based diagnostics, isothermal amplification, next generation sequencing, multi-omics and AI based diagnostics. The advantages are rapid detection, high precision, suitable for point of care testing. Now we have already learnt about the histopathological features. There are distinct hallmark features for each Epstein-Barr viral associated tumors. We have already discussed about the RSLs, the starry sky appearance, etc. Now let's come to the infectious mononucleosis. This is also caused by Epstein-Barr virus, but this is not a tumor, as we have already discussed. This is not a tumor like the other diseases we have already discussed. It is caused by Epstein-Barr virus, and it spread via saliva or in kissing or sharing utensils or drinks. This is also known as kissing disease. The virus infects the oropharyngeal epithelial cells and the B lymphocytes. The clinical symptoms are two types, like classical triad symptoms and other symptoms which are generalized. The classical triad includes fever, sore throat, which is due to pharyngitis, and lymphadenopathy, especially where the cervical lymph nodes are involved. And the other symptoms are fatigue, splenomegaly, hepatomegaly, and rash. The rash is seen especially after the use of antibiotic ampicillin. Now let's come to the pathogenesis of infective mononucleosis. Epstein-Barr virus infects the B cells via CD21 receptor, and it causes the polyclonal B cell activation. The immune response, there is an immune response towards the Epstein-Barr virus, which lead to the activation of CD8 plus T cells. This lead to atypical lymphocytes. These are known as downy cells. These are seen in blood. Now let's see the laboratory diagnosis. The monospot test or heterophile antibody test is a rapid screening method. Epstein-Barr viral serology, including viral capsid antigen IgM, indicating the acute infection, viral capsid antigen IgG, indicating the past or current infection, and eBNA indicating the past infection. Now coming to the complete blood count. You can see lymphocytosis and atypical lymphocytes. Now, what are the complications associated with infective mononucleosis? The most important complication is splenic rupture, and hepatitis, airway obstruction due to tonsillar enlargement, and other neurological complications like encephalitis and Guillain-Barré syndrome are also seen here. The future directions. Ongoing research focuses on novel biomarkers and immunotherapy approaches for effective Epstein-Barr virus associated tumor treatments and prevention. With this, I conclude my seminar. Thank you.